Welcome to Friday iCANX Talks. My name is Lan Fu, and I'm a professor in physics from the Australian National University. And I'll be moderating the talks tonight. So join us tonight is Professor Liz-Marzán from the Center for Cooperative Research in Biomaterials in San Sebastian, Spain. And also join us tonight, the panel is Professor Jiaming Nan from Seoul National University, our iCANX host, Professor Liz-Marzán from Peking University, and also Dr. Sarah Abeld from International Iberian Nanotechnology Laboratory. So let me first introduce our speaker tonight, Professor Liz-Marzán, who is an Iber-Basque research professor at the Center for Cooperative Research in Biomaterials in San Sebastian, Spain. He's also a group leader at the Biomedical Networking Center for Bioengineering, Biomaterials, and Nanomedicine. And he holds a part-time chair in physical chemistry at the University of Vigo. He has published more than 550 papers and is a co-inventor on 12 patents and has supervised over 40 PhD students and 60 postdocs, many of whom were professors worldwide. Professor Liz-Marzán's research has been recognized by many prestigious awards, which I don't have time to mention most of them, but I would really highlight his has a Humboldt Research Award in 2009, and also his inaugural ACS Nano Lectureship Award in 2012, and many more. He's also an international member of the National Academy of Engineering, USA, and a member of the Royal Academy of Science of Spain, the European Academy of Sciences, and Academia Europaea. He recently received an honorary doctorate from the University of Antwerp, Belgium. So without further ado, let's invite Professor Liz-Marzán to the stage. So please share your screen now. Yeah.
Okay, thank you very much. All right, well, thank you very much for the invitation to join the iCANX talks. I'm gonna try to give you an overview of research that we have done over the past few years, in particular related to trying to contribute to the translation of nanoplasmonics to what we call personalized medicine. So I'm gonna start by introducing plasmonics and nanoplasmonics, and hopefully in the end I will have enough reasons to conclude with describing what we call personalized medicine. So plasmonics is a field that has really exploded during the past, say, 2 decades, mainly driven by plenty of potential applications, as you can see here in these examples that I have labeled in this slide. Perhaps one of the early definitions of plasmonics was given by Professor Harry Atwater, and there was this popular article in Scientific American. And we could say that plasmonics is the field where we study the interaction of light with objects that are small compared to the wavelength of light, which is something which sounds quite counterintuitive, because normally the interaction of electromagnetic waves with matter is most efficient when there is a match between the wavelength of the radiation and the size of the object. If you think, for example, about X-ray diffraction, the wavelength of X-rays coincides roughly with interatomic distances, and this is why we use it to study atomic lattices. If you think about photonic crystals, then you need wavelengths that are matched with the lattice of micron-sized particles, so that you get, again, a good interaction. In plasmonics, you have something which is unusual, where it is a very tiny object which is able to manipulate and to affect the transmission and reflection of light. So how does this work? You can mainly look initially at what happens in the bulk. When you have materials with free electrons, for example, most metals, if you are irradiating with the appropriate electromagnetic radiation at what we call the bulk plasma frequency, there will be a translation of charge density waves through the metal, which are in resonance with the oscillation frequency of the radiation. However, if you reduce the dimensions of the metal, create a very thin film, then these waves will not be able to move inside of the metal, so they are confined at the interface between the metal and the dielectric material that is in contact with it. And this is what we call surface plasmon resonances, which are the basis for the commercially available surface plasmon resonance biosensors, meaning that you're evaluating the absorption of certain molecules close to the metallic surface, which will change, because of changes in the refractive index, the oscillation frequency of these waves. In very tiny particles, where you are reducing the dimensions to the nanometer scale in 3 directions of space, then you don't have enough area for these waves to translate anymore. And so typically, if you think about the most simple example, where you have spherical particles that are very small compared to the wavelength of light, then the electric field of the radiation will create a separation of charges, an electric dipole. And the equilibrium between this electric field and the restoring force for these charges to go back to equilibrium will result in another resonance frequency, which we will call localized surface plasmon resonance frequency. And this is what determines the colors of colloidal dispersions of metals, for example, gold and silver, where if you have small spherical particles dispersed, for example, in water, you will see that the dispersion of silver looks yellow, the dispersion of gold looks red. And so this comes from these very sharp absorption bands that are located in the visible, very unusual for metals. But this happens when you are in the nanometer regime. So silver will interact, or small silver particles will interact with light mainly around 400 nanometers. Gold particles will interact with light mainly around 500 nanometers, and this determines the different colors. So this is quite interesting, and it is even the origin of many stained glasses that we can see in medieval churches or even in Roman objects. So this was known for a long time, even though the reason was definitely not known. Now, another important consequence of this very strong interaction of light with small metal particles is that when the radiation is coming and interacting with the particle, it also induces an enhancement of the electric field right at the surface of the particle, which decays very quickly away from the surface. So just a few nanometers away from the surface, there will be no enhancement of the electric field. And this is a concept that I would like you to remember, because it will be the basis of the main technique that I'm going to describe during this lecture as a tool to go in the direction of personalized medicine. Now, I explained all these effects talking about small particles, but actually it's much more interesting if we focus on changes on the morphology of the particles. So, for example, if you have elongated particles like these nanorods that we call here, then the interaction of light with the particles can be different depending on the direction of the incidence of light and the polarization of the light. So you can have light propagating in this direction with this polarization, and then you will induce oscillation of electrons along the main axis of the nanorod. But if the nanorod is perpendicular to the polarization of light, then you will have oscillations across the main axis of the nanorod. And this also translates into differences both in the intensity and in the distribution of the electric field enhancement on the very same particle. So these are calculated maps. I'll tell you in a minute how this is done, where you can see that you can enhance very much the electric field at the tips when the polarization of light is parallel to the main axis of the rod, but you're enhancing the field in a lower extent and mainly at the sides when the polarization is perpendicular. And so because of these two main interactions of light with the elongated particles, experimentally, we record two well-defined absorption bands, one at lower wavelengths, which is related to this oscillation across the short axis of the rod, and another much more intense, which is related to the oscillation along the main axis of the rod. And it's particularly interesting in this case because the degree of elongation of the particles determines the position of this band. So what we call the aspect ratio, meaning the ratio between the length and the width of the particles, will be the main reason for the main localized surface plasmon resonance to be located either in the visible, mainly in the red, or all the way into the near infrared. And this is actually quite interesting for many applications. I was actually going to show you that by using colloid chemistry, we are now able to prepare gold particles or silver particles with many different morphologies, even in many different sizes. And the techniques that we use are so precise that in every case, doing things right, we are able to obtain particles that look almost exactly the same. And this is extremely important because if you remember, I just said that the position of the localized surface plasmon resonance is critically dependent on the anisotropy of the particles. And so if you have a mixture of shapes or a mixture of sizes, you will get a much broader response in the interaction of light with these particles, and therefore the efficiency of the process will be largely decreased. And so it's important to know how to make the particles. You have a few references here. There are quite a few other groups all around the world which are doing an extremely good job to prepare particles of very high quality. And each of these morphologies will have a different advantage for a particular application. Now, in the case of bio-related applications, we typically try to go to morphologies that allow us to have an intense plasmon resonance in this wavelength window, what we call the optical transparency window for tissue. In fact, you can define up to 3 or even 4 windows if you go farther into the infrared. But if you want to stay close to the near infrared, typically the window between around 700 and 950 nanometers is the most appropriate one for deeper penetration of light and at the same time minimizing the damage that the radiation can do on living cells. And so you can achieve particles responding in this wavelength range by choosing different morphologies, for example, triangles, what we call nanostars or nanorods, with the appropriate size and the appropriate anisotropy. And so again, I insist it's important to be able to dominate the synthetic methods in order to obtain the properties that will be most efficient for the application that you have in mind. And once you know how to make the particles, then you can start thinking what are the problems that you will like to resolve and how you are going to resolve them. Now, I wanted to give you also a little bit of the historical perspective of the technique that we are using, which some people call surface-enhanced Raman scattering, some other people surface-enhanced Raman spectroscopy, and the nickname is usually SERS or SERS, and it can be used for both of the names that we are giving here. They denote the same thing. And next year we will be celebrating the 50th anniversary of the quoted discovery of this effect. So most people assign the discovery of surface-enhanced Raman to this manuscript published by the group of Martin Fleischmann in Southampton, where they actually describe for the first time spectra of pyridine molecules which were absorbed at the silver electrode. In fact, if you look at the paper, there is not really a claim that they see enhanced signals of Raman scattering, but they do see changes when changing the electric potential on the electron. And so because of the existence of this work, there were other people who were looking more closely at this process. And so there are 2 other papers which are also quoted, like the origin of surface-enhanced Raman scattering. one by Albrecht and Crichton, where they studied in very much detail the intense Raman spectra of pyridine. Here there is already a claim of increased intensity in the signal because of the molecules being adsorbed on the metallic surface. And then this other by the group of Van Duyn, where they really proposed for the first time that the electric field enhancement would be the reason for the high signal in Raman scattering that was being recorded. And then from this place on, I probably should also quote Professor Moskowitz, who was the first to really describe the plasmonic relationship between the electric field enhancement and the Raman signal. But then from there on, there has been an explosion of research both on understanding this process. It's not so simple as it looks, because there are several potential mechanisms behind the reason of this enhancement of the Raman signal when it is adsorbed on a metal surface, but also on looking for applications. And we can see hundreds or even thousands of papers every year published on applications of surface-enhanced Raman. In fact, an example is this big review article that we recently published in our favorite journal, ACS Nano, where we collected many of the main authors in the field, collecting the present state of the art of the technique, the concepts and the applications, and also many prospects for future application of this technique, which is still bringing surprises and new fields, as I say, for application. So this is the main topic of my lecture, how to apply surface-enhanced Raman to help medicine. Now, if you think of surface-enhanced Raman scattering as an analytical tool, you can find many reasons to use it, many advantages. Selectivity, because Raman scattering is really a very selective technique. Every molecule will have a so-called spectroscopic fingerprint with well-defined peaks that will tell you exactly which molecule you're looking at. Sensitivity can be extremely high if you optimize your process, and there are several reports in the literature where single molecule detection is claimed. Again, this is usually done at rather specific setups and conditions. But you can also do very fast measurements. You can use portable systems. Sample preparation usually is extremely easy. And I'll show you examples of how you can use also this technique to prepare codes with a very large library so that you can think about applications in imaging or in safety. Among many other things, as you can see here, application is very general because most molecules will have activity in Raman scattering. Now, there are quite a few other issues that you have to take into account in order to bring this technique really to the market. One is the reproducibility of preparation of the substrates, but also reproducibility of the measurements. It's not so simple to prepare the plasmonic substrates that will give you always the same response. Even within the same substrate, if you're not careful enough, the uniformity may not be all the same everywhere. Another very important point, if you remember, I said that for this electric field to be enhanced, you have to be very close to the surface of the metal. And so the analyte has to come really almost to contact with the metallic surface in order to feel the high electric, the electric field enhancement, and therefore to give sufficient signal in surface-enhanced Raman. This is particularly difficult in complex fluid, for example, in biofluids, where you can have many other molecules that can interfere also with your measurement. And something that is also very important if you want to do measurements online over time, you can have memory effects, meaning that molecules can adsorb to your substrate, provide the signal, but then not leave and therefore still contribute to the signal that you measure at a later time. So I'm trying to tell you a little bit about how we have been working on trying to resolve all of these issues and come into a workable product. Now, substrates. As I mentioned, initially people were working with rough electrodes. Then there has been quite a bit of activity on the preparation of substrates, which are much better defined. The quality of the substrate is actually very important because this equation here tells you that the enhancement of the Raman signal compared to the standard Raman scales like the fourth power of the enhancement of the local electric field compared to the external field. And therefore, if you're able to enhance the local field in, say, a couple of orders of magnitude, you may be able to obtain 8 orders of magnitude enhancement in the Raman signal, which already will take you close to the single molecule detection. And therefore, preparation of the substrate is very important, and understanding the reasons why you can optimize the enhancement of the electric field will also be very important to know. And so there are, I think, 2 main effects that you can play with. One is the morphology of the particles. So I mentioned how by irradiating light with a certain polarization on a spherical particle, you can enhance the electric field at the 2 sides, and then it decays very quickly. If you are using particles that contain, for example, sharp tips, then it is known both experimentally and through theoretical modeling that you can have a much larger enhancement at the tips of the particles, which is very nice because you can get a large enhancement here, but it also poses some challenges because the molecule that you're going to measure has to come precisely to this spot, which is not always so easy to achieve. Another way of enlarging or optimizing the enhancement of the electric field is bringing particles close to each other. And so you're creating what we call hotspots. And I like to show this similarity with this other paper that was recently published by a group in Canada, where they actually show that you can also form hotspots if you irradiate bigger objects like 2 grapes, which are in contact, when you put them inside of your home microwave oven. So irradiating with a long wavelength on larger objects, you can obtain an effect that is similar to what you obtain here for very small particles, irradiating with visible or near infrared light, obtaining a very large enhancement of the electric field exactly at the gap between the particles. And so again, for this strategy to work, you need your molecules to come and sit right at the gap between the 2 particles, or more particles if you can create them. So this is roughly the 2 main strategies that you can follow to optimize the Raman signal, and I'm going to show you examples on how to do both of them. Now, before going into real examples, let me show you another prediction from theory, which I think correlates quite well with what we also see experimentally. Now, there are techniques that allow you to resolve Maxwell's equations using numerical methods. Similar to the maps that I showed you earlier, you can find ways to use these numerical methods to calculate the enhancement of Raman signal on very large sets of particles. And so our colleagues here in Barcelona, Vigo, and Cáceres were able to calculate the enhancement of the Raman signal for a large array of nanorods, randomly organized, similar to what we get in electron microscopy images. And by calculating this, they assign this white color to the areas where you can get a large enhancement, and the black ones to the areas which are completely silent. If you look carefully at this map, you will see that most of the signal that you will be able to record from an assembly like this will come from a very small number of hotspots. And therefore, you're really wasting a lot of material and a lot of area on your substrate. It still works, but it definitely can be optimized. Even more important, if you are using nanostars, the ones that I mentioned are extremely efficient because they have these sharp tips. It turns out that if you increase gradually the density of nanostars on a flat surface, they will start to interact with each other to the point that you can even switch off the hotspots. So you may be at a better condition with a lower density of nanoparticles than with a larger one, where you can get what we could call destructive interference between the plasmon resonances of nanostars that are touching each other in the wrong way. And so if you plot the enhancement of the Raman signal as a function of the density of particles on the surface, for spheres or rods, you see a gradual increase You're of course increasing the number of particles that are in your measurement spot. And at a certain point, you see an increase in the slope of the curve, because now you start to form the hotspots that I mentioned. So you get more of these spots that become white in the calculation that will give you the very large Raman signal in your measurement. For nanostars, it's not too bad, as I mentioned, but actually you don't gain much by increasing the number of particles. And so you're really, again, wasting material if you're increasing the coverage density, but you're not getting an enlargement in the Raman signal. So you should be aware of all these effects before you design your substrate for doing the measurements. Now, I come to the first application, or maybe the first big application that we had in mind, which was related to applying surface-enhanced Raman to study the communication between bacteria. So probably many of you are not familiar with this process. This is a mechanism that bacteria use to talk to each other, which is called quorum sensing, in which bacteria release certain metabolites that are then recognized by some proteins that are located at the surface of other bacteria. And then depending on the concentration of the recognition molecules that they can detect, they will know whether there are enough individuals in the colony so that they can activate mechanisms such as the formation of biofilms. And so usually to study this communication and this potentially threatening activity of bacteria, the traditional methods are quite invasive and quite slow. So what we proposed was, can we prepare substrates where the bacteria will happily grow, and then we can detect in real time the segregation of these metabolites, and then decide exactly when is the start of communication. How can we predict the communication between bacteria? Can we then take measure to control it? And so, again, from calculations, we decided that a structure like this would be extremely efficient, preparing a multi-layer of well-ordered gold nanorods. So as I mentioned, the simulations that you can see here are calculations of the electric field going down in depth in this tower of nanorods. And you see that periodically there is a formation of bright spots. These are the planes in between every two monolayers. So if the molecules are able to diffuse through the monolayers and get to these hotspots, then we should be able to detect a very low concentration of the metabolite molecules. And indeed, we were able to prepare something that looks very much like the model. So what you see here is a three-dimensional reconstruction of a multi-layer of gold nanorods. I wouldn't say it's a perfect crystal, but it's close to that. And we were able to do it in a way that we could open pores that would allow the diffusion of small molecules through the layers between nanorods. And in this way, we were excluding larger molecules available in the biofluid and only allowing the diffusion of the small metabolites. So this is how the substrates look like. And this is how we envision the experiment, where the bacteria, which are being cultured directly on top of these covered towers of nanorods covered with mesoporous silica, they would sit there, start doing their thing, and exchanging metabolites. And then by irradiation with a laser beam at very low intensity, we would be able to record the search signal, which are the spectra that you see here on top. And we are able to monitor the increase in the population of bacteria already at very early stages of incubation. And so this allowed us to do further work to study how different types of bacteria interact with each other and to really learn more about these important processes. One potential application of this detection system is to do this within animals. In this experiment, it was a dead mouse, but in principle, you can also think of doing this in living systems because the gold nanoparticles are quite biocompatible. And so by implanting one of these sensors behind the skin of the mouse, we were able to detect sub-micromolar concentrations of this molecule, pyocyanin, which is involved in the communication of one of the most devastating bacteria, Pseudomonas aeruginosa, causing a lot of trouble, especially in hospital environments. And therefore, you could think of using this system to monitor potential infections after surgery, for example. So now this was a project that we finalized a few years ago, and then we decided to switch a little bit field and go into the field of cancer, because there are many more challenges here. I wouldn't say it's a more important problem than bacterial infections, but it is again still one of the major causes of death worldwide. And so what we proposed is to use, again, surface-enhanced Raman substrates of different types to detect specific processes that take place in tumor environments, for example, tumor metabolism. And at the same time, we also propose to do this in 3D, to culture cells using scaffolds, and then to be able to label the cells with what I mentioned earlier, Raman encoded nanoparticles, so that we can also monitor the behavior of the cells within the tissue in real time. So this was a project that started roughly 5 years ago. So we have roughly 1 more year before the funding is gone. But I think we have progressed quite a bit, and this is what I would like to tell you today. Now, the part related to tumor metabolism was mainly driven by my student, Javier Plo. He's now a postdoc at a neighboring institute. And he was co-supervised by Professor Arcaiz Carracedo, who is a cancer metabolism expert at CIC Biomaterials. And so by initial discussions, we thought, well, it is well known that in the tumor microenvironment, there will be many molecules that will be indicative of the progression of the tumor. And many of these are related to the metabolism, which can be completely altered in the presence of tumor cells. For example, there are different enzymes that can be overexpressed in cancer cells, which are not in normal cells. So these will definitely change, for example, the conversion of tryptophan into kynurenine, the consumption of arginine, or the conversion of glucose. So we decided to start working on the design of surface-enhanced Raman scattering sensors to detect the altered metabolism of cancer cells. And in fact, some other direction that was coming up also almost at the same time was trying to identify cell death mechanisms. This is again related to the release of different metabolites. And depending on what you can identify after the death of the cells, you can also try to distinguish different cell death mechanisms. So we focus mainly on what is called regulated cell death, but depending on the reasons for inducing cell death, here you have many examples of potential external stimuli that can lead to cell death. What we thought is maybe by looking at the different metabolites, we can decide, for example, to distinguish apoptotic cell death mechanism compared to necrotic one. And so the idea is that we can culture cells, we can apply this external stimuli, and then in situ or extracting the supernatant from the cell culture, we can then measure the presence of different metabolites and even extract a pattern that will be able to tell us whether the secretion of these metabolites after cell death will become anti-inflammatory or pro-inflammatory, which is definitely important in terms of how to treat the cancer in the patient. Okay. All right, so this is the first piece of work that we did. As you can see, all quite early work, quite recent work. And so for this initial work, we actually took a lot of care to prepare substrates that would be very efficient and also very uniform. And to do this, we used some templates to induce the aggregation of gold nanoparticles into small clusters that would look something like this, which are ordered in 2D on a flat surface. And so by inducing this aggregation into these tiny clusters, which are separated from each other by a few hundred of nanometers, then we can couple the plasmonic interaction of light with the metal particles within each of these tiny clusters and the diffraction of light because of the separation distance between the clusters. This is what I mentioned in the beginning. Typically, you have efficient interaction of radiation for objects that have the dimensions comparable to the wavelength of light. This is what happens in diffraction. And then you can couple it with plasmonics, which has the unusual feature that light will interact very strongly with particles that are very small compared to the wavelength of light. And so by doing this, and simply by changing the lattice parameter, we can select the position at which the interaction of light will be more efficient with the substrate, and then we will also get the most efficient hotspots between the particles forming these tiny clusters. And so after preparation and characterization of these substrates, we started to think of metabolic processes. And so one of these processes, as I mentioned, is the consumption of tryptophan to form, of kynurenine to form tryptophan. And then this is actually obtained by an enzyme that is called IDO1, which is overexpressed in many cancer cells and specifically in HeLa cells. And so first thing we did was to check the possibility of distinguishing kynurenine and tryptophan by measuring the Raman spectra. And indeed, we found that there are certain areas where there are characteristic peaks that can be used to distinguish one metabolite from the other. And so this is a comparison between the spectrum of kynurenine on a substrate at very low concentration with the pure molecule and the standard Raman scattering signal. And the same thing for tryptophan. So you can see that the detection is actually quite clear. Now, what we did then was to incubate cancer cells. In this case, it was the standard HeLa cells. And then extracting supernatants from different cell wells, we were studying this process of conversion of tryptophan into kynurenine in real time. And so you can see that if you measure the spectra at different times by using always different substrates, you see that the peak of tryptophan is gradually decreasing, the peak characteristic of kynurenine is gradually increasing. Another feature that we observed by doing this experiment was that apart from these changes in the peaks of tryptophan and kynurenine, there was another peak that was very clearly increasing over time, and we found that this was characteristic of cell death. So indeed, by inducing the cell death by the addition of hemine, which is causing cell stress, then we obtained very clearly the formation of this peak, which we could assign to purine derivatives. And so indeed, we were able to control cell death induced by different processes, which was consistently related to the formation of this peak, which was associated with the characteristic band at 727, which we, as I said, assigned to these derivatives that they are typically released during most of the cell death mechanisms. So now then decided to work on this process a little bit more. For example, we could do the detection of this characteristic peak with local resolution. For example, if you have a very dense aggregate of cancer cells and you're gradually inducing cell death starting from the inside of the tumor, you see that as cell death is progressing, you're increasing the intensity of the peak that you're detecting. So you can really monitor this process in time and also with localized space resolution. Now, this is quite interesting and it's giving quite good results, but it has one technical problem. As I mentioned a few slides ago, if you have one surface-enhanced Raman scattering substrate, metallic array of gold nanoparticles, and then you expose it to an analyte that is absorbing on the surface of the metal, this analyte can absorb in an irreversible way. And then, for example, here, this is an example with standard Raman active molecules. We expose this array of gold nanoparticles to MBA, mercaptobenzoic acid, and then we record the characteristic peak, which is typically very intense around here, close to 1100 centimeter minus 1. And then we clean the substrate, we incubate in a solution of nicotinamide, which we expect to have a peak here closer to 1000 centimeter minus 1. But what we measure is exactly the same spectrum as we had before. If we clean the substrate and incubate it in a mixture of both molecules, again, we see only the peak of MBA, which means that the molecules that were attaching initially at the early stages are not leaving the surface, and therefore the new upcoming molecules are not able to anchor and provide the Raman signal. And so for all these experiments, what we had to do was to locate the Raman active substrate on top of the cell culture to measure directly the supernatant from a laser beam coming from the top, and then bringing a newly prepared substrate and place it very carefully on top of the cell culture so that it would not be disturbed, which, as you can imagine, is not only very cumbersome, it may potentially induce changes in the cell culture, but additionally is extremely expensive in terms of material and also in terms of time. And therefore, Javier and another student, Matthias, came up with the idea of using a substrate where we could open windows every time we want to measure. So the idea is that you have this array of nanoparticles. You can still see the white spots reflecting the position of the metal nanoparticle clusters, but now they are uniformly covered by a layer of PLGA, polylactic glycolic acid. This PLGA layer can be degraded when you increase temperature. And the metal particles are known to lead to local high temperatures when they are irradiated with a high intensity laser beam. And so the idea was, if we have this substrate and then we illuminate with the same laser beam that we use for the measurements, but at a slightly higher intensity, we can open windows. You can see here, this is an example of how locally you are increasing the temperature. This is an image from a thermal camera. So locally, you're increasing temperature with the laser beam. This results in this hole in the polymer layer, but the metal particles are still alive and happy. And now they are available for molecules that are coming from the solvent and attaching on the surface of the particles, but cannot reach the rest of the substrate. And so now you can do the measurement on this window that you have opened on the substrate. And so now you can very easily open one window, measure one chemical, get one spectrum, then you wash it, you come with a different solution, open a window at a different spot, measure whatever is present in the new solution, get a different spectrum, and so on and so forth. You can do this for hundreds or even thousands of times with the very same substrate by simply using a micrometric stage. And therefore, we also show that this can be applied to measuring cell cultures in real time. So you can really position these substrates on top of your cell culture, open windows every time you want to measure, and then you can see exactly the same results by using the single substrate opening different windows and by using different substrates and taking samples every time you want to do the measurement. So this is something that actually helps us to increase a lot the throughput of the measurements that we want to do. Now, coming back to cell death mechanisms, this is something that we continued studying, but as I mentioned earlier, so far what we were doing was to monitor this peak, that 727, that was characteristic of the purine derivatives that were being released during cell death. However, if you think of different cell death mechanisms, it's very likely that they will actually bring about different compositions in the supernatant after release of these metabolites or this cell content during cell death mechanisms. Not only that, even the death rate may be different depending on the stimuli you use for killing the cells. For example, if you use UV light irradiation, UV will slowly induce damage in the DNA of the cells, and this damaged DNA will guide the cell death, which will take a longer time than, for example, if you add hydrogen peroxide, which will disturb the cell membrane and immediately lead to a burst or release of the content of the cell. And so what we tried to do was to employ methodologies to analyze the full spectrum and not only one peak that is characteristic of one of the metabolites that may be present in the system. And so we started planning a process that starts by preparing substrates, acquiring a large number of spectra, pre-processing, and then classification by using machine learning techniques. In fact, initially we tried starting by standard PCA analysis. So what you can see here are average spectra from many thousands of measurements that you can see here all collected for the release of the content of cells after inducing cell death with different stimuli. So this is the control where the cells were dying naturally. UV light irradiation, heating the system, or hypotonic, where we basically add pure water, which has no oxygen, no salts and no food for the cells. And so the different mechanisms, as you can see here, lead to different signals in the Raman spectrum. And now, because we have a large number of data, then we start to do principal component analysis, and we can see already a segregation of families that basically correspond to the different mechanisms that we have here. Now, you can still see that if you compare now the segregation of signals for different mechanisms, you see, for example, that hypotonic is very well separated compared to heating and UV, but also that UV is closer to the control. And this is probably related to what I mentioned of the slower character of cell death by inducing DNA damage. Now, you can do the same type of experiment by collecting spectra at different times. As I mentioned, we can do that now at a very high throughput. We can obtain enough measurements so that you can again do classification at the different stages. In this case, we used T-distributed stochastic neighbor embedding method, which is more efficient at clustering the signals. And here you can see again that at shorter times, 1 hour, there is already segregation of hypotonic. It seems to go faster than the other mechanisms. And then at 4 hours, there is already better segregation of all the signals, but UV is still closer to the control. After 24 hours, you see a very good separation of the 4 types of cell death. And so now we can try to assign these to different mechanisms and classify. And so for classification, again, we started using non-supervised methods where we take the thousands of spectra that we have collected. We label them by using analysis of cytotoxicity by the LDH assay. So we can know exactly which spectra correspond to dead cells and which correspond to live cells by a certain threshold. And then we can train the machine learning algorithm so that it can be used then to classify another set of spectra. And so by using this method, we obtain an extremely good fit between what we know is the ground truth and what is provided by the machine learning model. We can also generalize this method by applying the same trained algorithm to now identify cell death mechanisms with different stimuli, with different substrates, and at different times. And so this is actually a method that we are starting to use much more generally. Another consequence is that you can measure not only the distinguishing death and live cells, you can start to think, as I mentioned earlier, about distinguishing cell death mechanisms. So again, by doing flow cytometry and applying standard labels for apoptosis and necrosis, we can again label the spectra coming from different experiments where we use different stimuli. And now we can again train the machine learning algorithm, do the validation of the method. As you can see, the accuracy is close to 100%. And we can also identify which are the main peaks which can be correlated to either necrosis or apoptosis. And by doing this, you can also use this process to validate by using flow cytometry, and you can even start to think about biomarker discovery, because now you can try to assign peaks from the spectra that are related to the different mechanisms, which are characteristic of some standard molecules, for example, adenosine triphosphate. This was identified mainly after applying the heating, so mainly leading to apoptosis mechanisms and necrosis mechanisms. And this is something that can open up new investigations on how to induce different processes in tumor cells. All right, now I change a little bit the direction. I mentioned earlier that we also wanted to use codes based on surface-enhanced Raman to identify the distribution of different cells, ideally in 3D. And so the preparation of these labels is actually quite simple. We start, as I mentioned earlier, by preparing the metal particles that we like, colloidal dispersions, rather monodispersed with exactly the optical response that we are interested in. And then by doing ligand exchange, we can deposit on the surface of the particles what we call Raman reporters, which are molecules that are quite efficient at scattering Raman, and they can attach easily to the metal surface. In this way, we can obtain a sort of Raman barcode that will tell us exactly which particle we are looking at. Because these molecules are close to the surface, the signal will always be enhanced, but we need to protect the surface with an external coverage so that no other molecules will come, bind to the surface and contaminate the signal. And at the same time, we protect the initial Raman reporters from leaching out from the surface. Additionally, on this coverage, we can even deposit targeting molecules like amino acids, so we can do this labeling much more selective. So an example of labeled particles would be something like this. You have the gold nanostars. You attach on the surface of the nanostars a hydrophobic Raman-active thiolated molecule. So this molecule is multi-aromatic and therefore is very active in Raman. It's hydrophobic, so the particles after modification will be dispersed in non-polar solvents. And it contains a thiol group so that the molecules will easily attach to the metal. After doing that, by mixing with an amphiphilic molecule that can self-assemble on the surface of the hydrophobic molecules on the particles, then you obtain a layer that is protecting the Raman reporter, and at the same time, because it's hydrophilic on the outside, it will make the particles also easily dispersed in biofluids. And by measuring the Raman spectrum of these particles, you can see there are very characteristic peaks that are being used as our barcode for the identification of these particles. And this is an example of how a cell would look like if you measure the characteristic positions of the peaks that are related to particles that have been internalized inside of the cells. Now, you can do that not only for one Raman reporter. So one of the advantages of this process is that the peaks are very narrow. So by simply changing a little bit the functional groups on the molecules that you're using to encode your nanoparticles, you immediately will obtain a different barcode, and therefore you can think of multiplexed labeling of different cell types. These are electron microscopy images after ultramicrotomy, which show that either nanostars or nanorods that have been labeled in this way are easily internalized, typically by endocytosis, and this is why you see them all grouped together in the endosomes that are formed by encapsulation of part of the cell membrane. Now, this is another example of multiplexing where we used 5 different cell types, which were labeled by internalization of gold nanostars with different Raman reporters. And here you can see that over time we were able to monitor the changes in the position of the molecules, which was not very much because this was a 2D culture. So they were really absorbing on the substrate, but you could still monitor over time, more than one day, how the position of the cells was changing by simply looking at these labels, which are additionally much more stable than fluorescent labels, so they do not bleach, and therefore you can also think of doing longer term measurements. Now, the extension of this work is the preparation of systems that become already 3D. And so one first example is this image here, or this couple of images, where you see this, where in blue we see the substrate, and then which here is actually placed on top. And then we have 2 layers of cells which were labeled by different target molecules. And then we actually can see that over time the different cell types are interdigitating with each other, and we can monitor the behavior of this system over time. Now, this is nice, but we really would like to go much farther than this. For example, we can design systems where the Raman reporter is additionally providing information about the local environment of the nanoparticles. An example is this pH sensing experiment. So we have the same gold nanostars. In this case, we use mercaptobenzoic acid as the Raman reporter because depending on pH, this carboxylic group will be protonated or not, and the signal will change. And therefore, you can monitor differences in pH if the particles are exposed to different solutions. In order to protect the molecules on the surface, in this case, we use polyarginine, mainly because it is positively charged and it actually helps internalizing the particles in most of the cell lines. And therefore, this is a calibration where we can measure the Raman signal of MBA as a function of pH. As you can see here, we can do on a rather wide pH range. And you see this peak is roughly staying constant, but this other peak here is being enhanced if you increase the pH value. And so now you can plot the ratio between the 2 bands, and then you can see that you can obtain a calibration curve that depends a little bit on whether the molecules are more or less exposed to the solvent. So depending on whether polyarginine has been deposited on the outside or not. And then you can also start doing measurements in a cyclic way using exactly the same particles, changing alternatively the pH of the solution. And you can see how you can determine different pH values cyclically, simply measuring the ratio and then using the calibration curve. So now you can think of internalizing the particles inside of cells. You can see here that by using polyarginine, the particles are getting much better internalized than, for example, using polyethylene glycol, which is known to be an antifouling agent. And now you can think of analyzing the changes in pH that take place during the internalization of the particles inside of the cells. And so the point is you have the extracellular region, you incorporate the nanoprobes, meaning the nanoparticles with the pH-sensitive Raman reporter. Then you wash, you wait for the particles to internalize, and you measure the Raman spectrum over time. And so this is an example of measurement in space resolution, where you can already start to measure the pH distribution. So this is a cell or actually 2 cells that were close to each other, where the nanoparticles were gradually getting internalized at different times. So first of all, you can see that at shorter times, most of the signal is coming from the outside. At longer times, you start to see accumulation more in the inside of the cells. And at the same time, you see that the color is changing slightly. So you can actually see how you're measuring a decrease in pH as the particles get internalized. Maybe you can see it better in this 3D slice. 2 hours, most of the signal is coming from the outside, from the membrane. The particles have not yet completely internalized. After 8 hours, most of the particles have been internalized inside of the cells, and therefore you see accumulation inside. And then if you analyze locally the pH from the Raman signal at different spots, either at the membrane or closer to the nucleus of the cell, you can see that at 2 hours, you see few particles have been internalized. Most of them are still at the membrane. At 8 hours, most of the particles are inside, and all of them have a lower pH, which is characteristic of the endosomes. So this is something that in principle you could also apply to cultures in 3D. And this is something that we are very much interested in doing, and we started doing already. So trying to increase the complexity of our cell models so that the analysis of all these processes becomes much more relevant to what really happens within a human body. So you probably, if you're familiar with biological studies, the most simple models are related to cultures or cell culture in 2D. This was most of what I have been showing you so far. Then you can create aggregates of particles like tumoroids or, for example, the layers of cells that I was showing you earlier. But you can also create scaffolds that guide the growth of cells in 3D. And ultimately, you would like to work with patient-derived scaffolds or biopsies that are obtained directly from patients who are suffering the disease. Depending on the complexity of the model, you will get a better comparison with the real thing. Depending on how, and the simple models will actually have problems, for example, related to diffusion of oxygen or nutrients, and then the access of drugs to cells that are aggregated will also be difficult. So what we decided to do was to go into an intermediate stage between the 2D cell culture and the animal models by employing 3D printing. So the main idea is we can 3D print gels, which are biocompatible, on which we can internalize the metal nanoparticles, which will be used as the substrate to enhance the Raman signal of the metabolites or other molecules that we want to measure. And at the same time, we can guide the growth of cells in 3D within the scaffold, and then we can also think of studying their distribution in 3D. And so this is how our first scaffolds look like. You can see we can print these lines. Every line is actually quite porous because it's formed of a hydrogel with mainly polymeric components. And then we can draw, different compartments with the holes where the cells will happily fit. And if you do microtomy again of one of these lines, you will find many of these particles which are internalized, separated from each other, providing individual signals of the molecules that will come close to their surface. And now we can come with the laser beam and try to analyze, for example, the signal of adenosine, which has been released by cells that are being cultured in this 3D system. So the signal of adenosine is labeled in green here. So you see that we can really get a signal from everywhere in the scaffold, meaning that these particles are uniformly distributed everywhere where we have printed. And at the same time, we can see the red color, which comes from an encoded nanoparticle that we have internalized inside of these cells. And so this is a first demonstration that we can really apply this system to create three-dimensional cell culture systems, which can be used to detect metabolic processes in real time. Now, let me show you another example of how this could be used, for example, to evaluate anti-cancer drug efficacy. So this is, again, the same type of scaffolds where we have the gold nanorods inside. And now what we are going to do is we are incubating cells inside of the holes in between the scaffold, looking like this. And then we are going to add in this tiny chip, we are going to add the anti-cancer drug here. And we are going to monitor the diffusion of the drug by the surface enhanced Raman scattering signal and correlate the diffusion of the drug with the death of the cancer cells. So as a model drug molecule, we selected methylene blue, mainly because it's much easier to control, because it's only an anti-cancer drug if it is photoactivated. And additionally, it features a rather high Raman signal, so it's very easy to monitor. In fact, what you see here is a 3D rendering of the scaffold that we are using, where the signal is coming from this peak that is characteristic of methylene blue. So we can, again, confirm that gold nanorods are dispersed everywhere in the hydrogel of the scaffold. And second, methylene blue is able to diffuse through the pores of the gel and then reach the nanoparticles where the Raman signal will be enhanced. Now, this is a very brief summary of the results. So first of all, we studied the diffusion of methylene blue through the scaffold containing Matrigel, which is a standard cell culture medium for 3D cell cultures. Because of the viscosity of Matrigel, the diffusion of the methylene blue is rather slow. I'm sorry I didn't put the times here. This is 1 hour, this is 2 hours, this is 24 hours. And so you can see that it takes quite a long time for the molecule to diffuse through the whole system. If you do the same experiment in the presence of the cells dispersed in the holes, as I showed you earlier by those green spots, then the diffusion rate is quite similar, but the intensity is lower because now a good part of the methylene blue is being internalized by the cells, which is exactly what we want, because then upon irradiation at 630 nanometers, then we can induce cell death and we can correlate. So cell death is here indicated by the propidium iodide signal in red, and the live cells are labeled by the green fluorescent protein signal in green. So you can see that there is again also a diffusion, a correlation in the cell death density when you go away from the reservoir where we injected the drug. It correlates very well with the diffusion rate of the drug that we can analyze by the Raman signal. So this is, as I said, an initial demonstration of the application of this system. Now, if we want to obtain more realistic models, then we would have to prepare something more similar to this. So these are studies from the literature which show a well-developed human tumor, where you see there is a core where there is a very high density of cancer cells, which is surrounded by different colors which reflect what is called the stroma. So the stroma is the environment of the tumor core, which contains different molecules, like collagen, for example, but also contains different cell types, like endothelial cells or cancer-related fibroblasts or cancer-associated fibroblasts, which are very important in the development of the tumor. So in order to obtain a realistic model, it's not enough to have the cancer cells. You should have the cancer cells at the right density and with the appropriate environment. So this is what we try to do also using 3D printing. So in this case...
We're running, I think you have 2 minutes. 2 minutes.
I'm able to finish in 2 minutes. That's fine.
Thank you.
All right. So first of all, what we tried to do was to obtain the most appropriate matrix to culture the cancer cells in a realistic environment. And so we started by obtaining breast tissue from dead pigs, from porks, and then developing a methodology to decellularize this tissue, extract all the animal-derived cells plus the DNA content. And then we were able to see that our cells were actually spreading very happily in this matrix. And by doing this, then we can think of establishing a model like this by using a 3D printer where with one piston we can print the high density of cancer cells in the middle, and with the other piston we can print something that looks like the stroma, containing collagen, but also containing the epithelial cells and the fibroblasts. And this is what you see here labeled in different colors. And then we can also, again, start to test the efficacy of different drugs in a much more realistic environment. And just to finish, I wanted to show you some very recent work that we are starting to do to show that even in this system that is not guided by a scaffold, we can also think of printing scaffolds. So after printing this model, we can use like a concentric piston where we can deposit within the realistic model, we can print cylinders that contain the nanoparticles inside. They somehow look like this. So this is a gel where we are printing these cylinders containing gold nanoparticles, and then we can start already recording the signal of molecules that are diffusing through the inside of these cylinders. And so with these tools, what we intend to do is try to help in the process of transitioning from the standard current medicine, where one type of therapy is being used for basically every type of patient, and with very different results depending on the condition and the genetics of the patient, into a medicine where we can do a previous analysis of the condition by using different techniques, segregating patient groups, and then applying the appropriate therapy. And so even though nowadays most of this is being done already by using DNA sequencing, what we would like is to implement surface-enhanced Raman as one of the standard techniques to actually analyze the status of the patient and the most appropriate drug to treat the disease. So with this, I finish. I'd like to thank the participation of many people in the group, collaborators, funding agencies, and thank you very much for your kind attention.
Thank you so much, Luis, for a really fascinating talk. It's really a multidisciplinary, combine so many exciting results. Now let me introduce our panel tonight. Okay, so our first panelist is Professor Jiaming Nam from Seoul National University. A brief introduction, Professor Nam is currently working as a professor at the Seoul National University. He did his PhD from Western University in 2004, and then he did postdoc fellowship in both UC Berkeley as well as Lawrence Berkeley National Laboratory from 2004 to 2005. Professor Nam's research is to develop a new generation of biosensors, therapeutic agents, or cell-based assays created from highly functional nanomaterial structures and biomaterials important in human disease and health. And of course, another panelist tonight is Alice, who doesn't need any introduction from me. Alice, of course, is from Peking University. And our ex-challenger tonight is Sarah Abelda from International Iberian Nanotechnology Laboratory. And Sarah is a Marie Curie co-found research fellow in this laboratory in Portugal. And she leads an interdisciplinary research project within the interface of colloids, microfluid droplet surface enhanced Brahman spectroscopy and cancer. And also the project aims to develop a novel and integrated platform for the analysis of cancer cells in biological fluids, in microfluidics and microdroplets. And also joining us tonight is our good old friend, Professor Nicholas Peppers from the University of Texas, Austin. Professor Peppers has a very long, distinguished career and with lots of awards and achievements. However, due to time, I would not have time to go provide the details. Let's straight away invite everyone to the stage. There we go. Okay. So as usual, let's start with Sarah, who as an ex-challenger of tonight. What's your question?
Thank you very much. It is so actually Luis was my professor while I was doing chemistry. So I met Luis when I was 19 and he taught me thermodynamics, and I always really liked his lectures, but I keep learning after almost 20 years after from Luis. So it is very overwhelming. I've written like 20 questions, so I'll probably have to then call Luis. So for the sake of time, I'm going to try to summarize it in 2-3 questions. So one of them is related to all the cell death monitoring that you did show, Luis. And so I tried to compile my questions, which is do you think that if you test that for several different types of cancers, you're going to be able to use the training algorithms that you have developed are you going to have the same labeling for necrosis and apoptosis, or do you think that there might be some differences?
Well, this is probably a bit early to tell. It's of course the future dream would be that you can apply the same training algorithm to then analyze the cell death in different cancer types and be able to use it as a diagnostic tool. I don't think we are there yet, but we are definitely working much more with this type of analysis tools to do discovery of new processes in cancer metabolism. We have just finalized a piece of work where we actually discover mechanisms by which certain cancer types are able to somehow escape the immune attack by consumption of a certain metabolite. Which is something that was not really new for the cancer biologists. And then we actually confirmed all these mechanisms by applying the standard techniques, which are usually much more time consuming than surface enhanced Raman. So I think that there is really a very big room to now exploit all the information that we have in the spectra that typically has not been very much exploited. And I see also many other colleagues who are using more and more these new AI-based tools to do the analysis of Raman signals. And I do think that if you do the data collection properly, one of the main problems is collecting so much data. You need a very high throughput system where the substrates are reliable. And as I said, this is still a bit of an issue. I didn't mention that in most of these high throughput analysis, we did not use anymore, or at least not all the time, the well-defined substrates, because the sensitivity that we had was enough if we were using drop-casted films of silver particles. And so, well, I may be deviating from the question, but even though you can use many types of substrates, by using simple ones, you can start generating much data because the signals are strong enough in very short measurement times. And then you will be able to train your algorithms and apply them where it is appropriate.
Thank you. That opens the way to my next question, which is sort of in between what you just said. So when you were explaining this thing of the cell death sensing, I wrote whether you were wanting to explore drug effects and how these drugs, may lead to different death mechanisms. And then you explained in these latest slides how you're doing that. But the way that you're exploring in these more complex 3D scaffolding models, the cell death is with traditional standard live death assays in fluorescence. So do you think that you can use your method based on SIRS in those models, as well as do you envision the establishment of SIRS as a standard technique to measure these and many other things? What is preventing SIRS to really make it through to the clinic?
Yeah, that's the key question. I think that's the reason why SIRS has not received the Nobel Prize yet. Well, I guess there are many reasons, but the crucial reason, and Nicholas will probably know more than me about this, the crucial reason is that you have to convince the clinicians that this is reliable. And so until we manage to do that, it will be impossible. So I know you are working also on this in this field. We are having a bit of trouble to convince people at the local hospital to provide the appropriate sample so that we can go into patient-derived tissue. And so we really need to team up very strongly. Interdisciplinarity will be key also, but we have to do a much better job into convincing the reliability of this technique. And this is why I think going into more complex systems where it's not so easy to work in general will be also a key step into demonstrating the reliability. Because then you can show we can obtain these results very quickly with rather simple methods. and they agree perfectly with what you do with gene analysis, proteomics, and so on. And so I think that will be extremely important.
Thank you, Luis.
Nicholas, I saw you turn on your microphone. Please go ahead if you have a question.
Luis, this is an incredibly good talk, and I hope that the students participated and I understood everything. As you very well said, the paper in ACS Nano from 2020 is a marvelous review of surface-enhanced Raman scattering. Like Sarah, I have a lot of questions, but I will not answer them. Instead, I will not ask them. Instead, I will focus on one that you brought up. As you know, as my age increases, I have become a patient. And I suffer from a number of diseases, prediabetes, and this, and that, and that. And when I go to the doctors, I don't see personalized medicine yet. Nobody mentions it for several reasons, one of which is the one you said. The medical doctors are a little bit worried, and the hospital does not give permission for those studies. But the other one is the financial problem. The financial problem is that our countries, in Spain, in China, in the United States, the companies, the insurance companies, I don't know what Korea will tell us, the insurance companies, they want to contain the costs. So you come in and say, you're not going to take this particular device or system or preparation that can treat your prediabetes and it is personalized because that's very expensive. Instead, we will give you the, I don't know, $1 Ozempic product that you will do every Friday evening. Is there any comment you can make as a fundamental scientist? We
have been only recently started to discuss with oncologists. The oncologists are reasonably positive, and they really would like to see new tools coming and helping them. But they also insist that if you come to the doctor and say, I have created this tissue from the sample you gave me from the patient, and I tested these 3 different drugs at different concentrations, and this is the right dose that you need to kill this cancer, they will never trust you, or at least not for the moment. And so I don't think, the cost is very high because the research is very expensive. And therefore, we know that also for any type of medicine, the pharma companies are investing a lot before they can get to a product. We mentioned already before the start of the talk that clinical trials are extremely expensive. And therefore, if they want to make profit, they have to charge a lot for the drugs. If it is something that is very novel and it has taken a lot of effort to fund, then the cost will definitely be higher, unless, as you said in the beginning, the governments get into it. And because most of the research is government funded, then there should be also a much better reach to these new techniques. I think that comparative treatment and comparative analysis should be a much faster way to go. I think we should use it much more often. But again, what I hear is extracting samples from the patients is painful, and the clinicians also want to have enough statistics for the analysis that they already do, and therefore it's not so easy to spare samples for research. So unless we come to an agreement on how to do that, it will be difficult to advance. But I know that many people are getting better agreements, and I hope we will also get there.
Our friend Patrick Ouvrer in France has been able to be getting all kinds of permissions do his nano studies,
That's right. Thank you.
Thank you.
Great. Professor Nam, do you have any questions?
Yeah. I think it was a very nice talk. You're going through all the fantastic application in biomedicine with the thirst. Luis, I think, as you know, Raman is fantastic because it can give you lots of information, right? The molecular fingerprint pick. It's actually really too complicated when you think about this non-specialist, right? For medical doctor, maybe fluorescence is easier, RGB, just simple emission pick. But Raman may be too complicated, right? But although it contains lots of information, especially when you apply this thing for like a complex fluid like blood, right? There will be lots of molecule. It's going to be even more complicated. So impossible to analyze that with your own eye and all these things. There are lots of uncertainty there. You need a huge enhancement, million times, even more than that, a lot of cases. So AI should, I think, come in and play a huge role in analyzing Raman signal. Actually, I have also experience. I did a lot of bio fluid analysis last 3-4 years. It actually really, really successful, especially when you start to incorporate this patient sample, for example, right? Now I analyze like for cancer diagnostics with the Raman, like thousands of patient sample. Actually, with the bigger data, and also and also the quality of these sample matters too, right? These clinicians, they need to understand what we're gonna do with this sample, right? So all the standard protocol, all these things also matter too. But problem for like this kind of AI based analysis is a lot of times you don't know why you come out with the result, right? It's a lot of times it's unexplainable. So I mean, in that regard, I mean, what do you think about this? Right? It's the paradigm for, I mean, you mentioned about biomarker. Maybe with the Raman, maybe the best way to, come up with a diagnostic system, maybe based on this Raman molecular fingerprint pick, rather than just one biomarker. That way, maybe it's more beneficial than, the kind of a conventional method. Maybe also that way you can convince the clinician Raman is maybe the only way to come up with a new types of a diagnosis system, right? That cannot be done with maybe typical fluorescence, right? So
yeah, well, as you very well know, and I tried to explain, one of the major advantages I think is in labeling. You will also developed a very, very extremely successful systems for encoded particles. And I think those really offer some advantages that fluorescent labeling does not. So one is the multiplexing, the possibility of getting images with one single laser line, and in particular the much longer lifetime of the measurements. On the other hand, I think certain Raman can be made as simple as fluorescence if you simply assign a color to a certain code, right? And so if you are providing directly the analysis, but you have to be sure, as you say, that the analysis is the correct one. In my opinion, at least for the initial application of AI tools, you really have to know what you are doing in terms of the assignment of the peaks to the certain vibrations in the molecule you are detecting. If you are, I really don't like the approaches where one takes a sample, measures a spectrum, introduces in the machine, and then obtains a reliability test without really knowing where the peaks are coming from. But if you are able to really know exactly what you expect, or more or less exactly what you expect, and then do the analysis and obtain a signal that correlates well with the assignment of peaks that you have for your molecule, then it's much more reliable.
Okay. Do you have any questions?
I just have maybe a follow-up comment question, which is it is on that very same line. I think that also Ceres and Raman people we might be a bit too judgmental on our technology because it's newer than fluorescence, but we all know that fluorescence antibodies coming from different companies or from different batches and lots, they need optimizations in the same way that these Ceres encoded nanoparticles would need, as any biosensor needs, an internal standard and calibration. So I guess that it is really the matter of showing the technology and convincing the oncologists. That would be my take. Of course, I'm a Raman and Seres apologist, but
Alice, do you have anything to add or ask?
Okay, yes, actually, Louis, that's really, really fantastic talk. Yeah, I learned a lot, especially I get very happy to see that micro and nano fabrications also work, right? Yeah, in your research, so that's quite encourageable. So I have a question for the last part you want to, for the 3D, you use, the printing, right? Yeah. You use printing. Did you think this was, this was the best way? Because, if you use all this 3D printing, that really costs a lot. Also, not mass production. Yeah.
Well, the materials that we use are actually very simple. We use standard biopolymers that can be formulated into hydrogels. So I don't think the material will be very costly, and the cost of 3D printers nowadays is really going down dramatically. You can even buy on Amazon very cheap printers and adapt them for the creation of this type of patterns. Of course, if you want to do it much more sophisticated in a sterile environment and by using UV light for hardening of the hydrogel, then it becomes a bit more expensive. There are definitely other ways of doing this, and there are many in the literature and probably even in the market which allow you to create these three-dimensional cell cultures. The issue is, first of all, if you want to incorporate the nanoparticle sensors, you need to know where they go. And you need to make sure that they are uniformly distributed in your scaffold or any other way of creating your 3D cell culture. If you don't want to do localized measurements, then you can use very simple techniques. Even pipetting might work to create a distribution of materials. But if you want to introduce sensors, you need to find a way that they stay at a certain spot so you know exactly where you have to measure. Otherwise, it will be much more complicated.
Okay, great. You mentioned about that, the sensors. So I saw in one example is detecting the pH, right? Yeah. So I saw as quite, large, kind of values. But in my mind, normally, in the body, the pH was not that large, difference, right?
No, but there are some reports that indicate that you may have also pH differences when you have tumor activity. So that was, again, what we had in mind, trying to see whether under certain stimuli, you say also changes in pH locally, where the cancer cells are doing a different activity than the normal cells. We are also trying to go, we are in a different direction, but related to this, we are also working on the incorporation of nanothermometers into these scaffolds, so we can also monitor metabolic activity by changes in temperature.
Okay, that's nice. Thank you.
Alice and Lan, if we have 1 minute, can I ask one simple question?
Go ahead. Sure. Yeah.
Luis, let's assume that the Spanish and the European government says, yes, you can use your technique. I'm interested in the tumors, the last part of your discussion. And let's say the medical doctors say, yes, we would like to study it. My question to you is, the technique cannot be used in vivo yet, can it be? Because there will be other parameters that will be affecting the different electrolytes, peptides, lipids, whatever.
In principle, it can. If you remember what I showed about the bacterial communication detection, you can implant a hydrogel containing nanoparticles inside of the body. And if the location is not very far from the skin, then the penetration of light will be sufficient to provide you with the signal that you need.
I saw that, yeah.
All right. I might take the opportunity to ask also a very tiny technical question, if I can. You mentioned very clearly that it's critical to control the shape and the size of the nanoparticles to achieve the best results. And it's very impressive, like when you do that, study the cell death, you do this regular alignment of nanoparticle with very accurate spacing nanometer scale. How could you do that? Can you just briefly introduce technique? Thank you.
Yes, I admit it was a bit fast when I described that one. So what we use is a pattern template, so a PDMS mold with tiny holes. And so by depositing this mold on top of a droplet containing the nanoparticles and then letting the solvent evaporate, they accumulate in the holes, and then you can peel it off, and you obtain this 2D distribution of the particles on the solid substrate.
But in between the molecule distance, how could they not agglomerate? Rather, they actually quite nicely with a nice sort of nanometer spacing.
Well, of course, initially the particles have some coating that also play in the interparticle interactions. And typically after preparing the substrate, if you want to obtain a very high sensitivity, what we do is to clean the organic material with plasma so that it's not anymore in there. But then the particles are already organized, so there will be no problem with that.
All right, great. Thank you very much. I think that's what we probably have the time for today. And with that, let me just, finishing the routine. So this is the certificate that I would like to represent iCANX to present to you, and Alice will deliver to your mailbox very soon. And thank you very much for joining us tonight for the iCANX talks. And next week, our speaker will be Professor Rebecca Kramer-Botiglo from Yale University, and she'll be talking about software robotics. So looking forward to that for next Friday, iCANX talks. And so with that, thank you very much, everyone, and see you next time.
不再是奇迹，不再是幻想，此刻真感觉全世界为我鼓掌。不必太在意身旁惊奇的目光，可以点点头，可以放声歌唱。我创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞。不再为曾经失败放弃或感伤，努力才是真的方向。I can， 没有什么可以阻挡心中无限的力量。